everybody, dear students. In the previous class I had explained you about different steps in cell cycle. Today our topic is mitosis cell cycle part 5. I am repeating, our topic is mitosis cell cycle part 5. What is meant by mitosis? Dear students, in the previous class of the cell cycle I had explained you about interphase, G1 phase, G2 phase, S phase. What is meant by interphase? It is a preparative phase, resting phase, all essential DNA, essential proteins, RNA, what are the requirements for mitosis, all essential criteria are going to be synthesized in the interphase. So, if you see an interphase, there is no cell division in the interphase, but there is an increase in the cell size. You understand? So, in the interphase, increase in the cell size, but there is no cell division. If you come to here, mitosis, it is an actual cell division. Actual cell division takes place in the mitosis. I am repeating, actual cell division takes place in the mitosis. I already explained about the human cell division, the generation time. Human cell will divide for every 24 hours, right? Out of this, more than 95% of time was consumed in the interphase only, hardly 1 hour for mitosis. So it is very short period where the cell division is going to be complete in a matter of 1 hour. So this mitosis is also called as karyokinesis. The meaning of karyokinesis means division of nucleus takes place in the mitosis. Division of nucleus takes place in the mitosis. Dear students, there is a question that S phase, so invisible phase of mitosis. In the previous class, I said that S phase is an invisible phase of mitosis. Why? Because the DNA replication starts in the S phase, synthetic phase, so that is called as invisible phase of mitosis. But in this case, the total nucleus is going to be separated. So division of nucleus is called as karyokinesis, that will be takes place in mitosis. Now one more point. If you see, mitosis is called as equational division. Equational division. What is the meaning of this equational division? Equally. What is that? Equally. If you see the example, if the parent cell has 2n, a diploid cell, whereas the cell is going to divide into 2 cells. In mitosis, one cell will divide into 2 cells. As a result, whatever the chromosome number present in the parent will be equally distributed to the 2 daughter cells. That is called as equational division. So, whatever the chromosome number present in the parent will be equally distributed to the 2 daughter cells without any exception, right? This is called as equational division. Now next point, there is no change in the chromosome number. Why I am using this word? Because in case of meiosis, a diploid cell subjected to meiosis, as a result 2 haploid cells are going to be generated. This is called as reductional division. For example, in case of meiosis, there is a 2 cell, a diploid cell. As a subjection of meiosis, haploid cells are going to be generated. Haploid cells are going to be generated. That is called the reductional division. But here I am repeating, no change in the chromosome number. Okay, that is the meaning of no change, means there is no decrease or increase in the chromosome number. That implies that no change in the chromosome number. Very important point that in case of plants, this mitosis takes place in both haploid and diploid cells in case of plants. In that concern, let me give an example in where the haploid cells do undergo mitosis. In the case of lower plants like Bryophyta, the gametophytic stage of Bryophyta is haploid condition. The gametophyte, the meaning of gametophyte means which will produces gametes, the male gamete and female gamete. Bryophyta are called as amphibians of plant kingdom. They are the lower level plants After the Thallophyta, the emerged plants are called as Bryophyta. These are habitated in both as well as soil as well as water areas. Means they require both water and the soil. They need water to complete their sexual reproduction. In this concern, these Bryophyta are the haplontic. They are haploid plants where they do undergo mitosis to produce the gametes. You understand? And as well as the diploid cells also participate in the mitotic division. You understand? I am repeating one more time. Plants do undergo mitosis both in haploid as well as in diploid cells. Example, for that haploid cell which will undergo mitosis is Bryophyta, which is the haploid condition of Bryophyta. Gametophyte stage is haploid. The gametophytic stage is haploid. The gametophyte means which will produce gametes via mitosis, not by meiosis, via mitosis. Next, in the case of animals, only diploid cell will do participate in mitosis. No haploid cell is going to be participate in mitosis. Only diploid cells only will participate in mitosis. Now coming to the different phases of mitosis: prophase, metaphase, anaphase and telophase. Here are the few points which will target the main issue in the prophase. Main target points in the prophase, metaphase, anaphase and telophase. If you come to the prophase, what exactly happened here is condensation of chromosome. What is meant by condensation of chromosome? Dear students, I explain you that DNA is thread-like structure in the interphase. The thread-like structure of DNA which is associated with the histone proteins are called as nucleosomes. That is called as chromatin. This chromatin is present in the case of interphase, whereas coming to the mitotic prophase, this interphase, the chromatin structure is going to be condensed to chromosome. So this is the structure of the chromosome. So a thread-like structure of DNA is going to be condensed to form a chromosome structure. This is called as condensation of chromosome. That's the first point. Second point, whatever the cell organelle is, endoplasmic reticulum, nuclear membrane, Golgi complex, nucleolus, all these cell organelles are going to be disappear in the prophase. Those are the two important points in the prophase. Next, coming to the metaphase. Dear student, metaphase is the phase where it's very actual phase to visualize the structure of chromosomes. In order to study the chromosome structure, metaphase is very nice phase to go through the structure of chromosomes. And not only that, congregation of chromosomes. What is the meaning of congregation? Congregation means whatever the chromosomes present in the cell all will align in the center part of the cell. Please see the diagram. So this is the cell. All the chromosomes will align in the center part of the cell. So chromosomes will align in the center part of the equator part of the cell. It is called as chromosomal congregation or metaphasic plate. That is the second point. Coming to the anaphase, very important point, dear students. Centromere. So this is the chromosome. The middle part, this is called as a primary construction. The primary construction is going to be cleaved into two parts. It's called as division of centromere takes place in the anaphase. I am repeating, division of centromere takes place in the anaphase of mitosis. It does not take place in the meiosis. No division of centromere takes place in meiosis. I am repeating, no division of centromere takes place in meiosis 1, not in the meiosis 2. Right? Meiosis 2 is equal to that of mitosis. So centromere will divide in the anaphase of the mitosis. Finally, the last point is whatever the cell organelles disappeared in the prophase are again going to appear in the telophase, last phase of mitosis. Telophase, endoplasmic reticulum, Golgi, nuclear membrane, nucleolus are going to be reappear in the phase which will complete the karyokinesis. So, in the next class, we will detailedly explain about each and every phase of mitosis, the prophase, metaphase, anaphase and telophase. Thank you.